Hello everyone. I'm John Looney from the University of Rochester. I'm a rheumatologist and a clinical immunologist, and I'm delighted to be here today at the Cleveland Clinic to talk to you about T-cell activation and control. This is part of a series that hopefully you've just completed Chapter 2 on with William Rigby talking about T-cell ontogeny. The learning objections for this particular section will be to be able to describe the role of T cells in other antigen-presenting cells in T-cell activation, and to identify and define signal 1 and signal 2 in terms of T-cell activation, explain the importance of co-inhibitory pathways for preventing adverse effects from immunological activation, to describe immunological exhaustion and how this may prevent control of chronic infections and malignancy, and as a sidelight to explain why we actually have CD4 and CD8 T cells. Many of the figures from this are from these books from Garland Science, particularly Chapter 9 in the Janeway text. So when you get infected with a virus, as shown here in this diagram, you can see that the virus titer climbs rapidly for the first few days, but then plateaus, and after about 5 to 6 days begins to fall back down. What prevents the viral titer from going up and allows it to plateau in those early few days is the innate immune system, where things like interferon alpha and beta are produced to slow down viral growth and replication, and NK cells are recruited to the site. NK cells are a type of lymphocyte that's part of the innate immune system and can be cytotoxic for virally infected cells. But what's really required to clear the viral infection is the activation of the adaptive immune system, where you have T cells coming in and eliminating those cells that are infected with virus. And this is when you have drop in the viral titer and elimination of the virus. So we want to talk today about the decision to activate T cells, and looking through some images for an analogy with this. Here's two images which are probably not very representative of the way T cell activation occurs. It's not a random event such as the flip of the coin depicted on the left. And it's not the T cell, here represented by Hamlet, thinking about whether he's going to respond or not. It's really more like this Walt Disney picture. You can see here there's a very handsome T cell being activated by an antigen-presenting cell. And several things are required. You have to have close contact from these two cell types. You also have to have specific recognition, or signal 1. And you have to have a positive signal given, which would be signal 2. And we're going to talk about the mechanisms of these two signals in the first part of this talk. In the second part of the talk, we're going to talk about controlling this activation. Obviously, if our T cell gets too activated, you have to have a mechanism for putting on a brake. And that will be discussed in the second half of this talk. Here are some actual antigen-presenting cells, and as you can see, they are quite beautiful, shown in the scanning electron micrographs at the bottom. There are dendritic cells, there are macrophages, and there are B cells. All of these cells are professional antigen-presenting cells and can activate T cells. The dendritic cells are particularly important in activating naive T cells, whereas both the macrophages and the B cells are very good at activating memory B cells once they've initially been activated by the dendritic cells. The dendritic cells are part of the innate immune system, and they are located in the sites where you are going to come in contact with microorganisms such as the skin, the respiratory mucosa, and the GI mucosa. And they're sentinels there, where they're in a resting state, waiting to interact with something that enters the system. Once they are engaged by a microorganism of some sort, they become activated. They migrate through the lymph to the lymphatics, And in the lymphatics, they interact with T cells and B cells and cause naive T and B cells to become activated. The activated cells then migrate through the lymphatics back to the bloodstream and then can home to the areas of inflammation where the microorganisms were originally entered the system. Just showing here, this is a blow-up of a lymph node. You can see the dendritic cell coming into the lymph node through the lymphatics at the top, and it migrates into the paracortical areas shown here in the light blue. There it's joined by the T cells and the B cells, which migrate into the lymph node via the high endothelial venules that are shown there as HEV. And what happens in the lymph node is that you have a constant progression of lymphocytes coming through the lymph node and sampling what's going on there. And if they interact with a dendritic cell that is activated in a certain way and carrying the antigen that they're specific for, then they are retained in the lymph node where they become activated and proliferate. And then after that activation and proliferation, they migrate into the lymphatics and then to the bloodstream. So there are two signals that need to come from the dendritic cell, signal 1 and signal 2. Signal 1 is this specific recognition, and signal 2 is the co-stimulatory or positive signal. This is a picture of the sort of the molecular biology that's involved in signal 1 and signal 2. Signal 1 is an interaction between the T cell antigen receptor, shown there in blue on the T cell, that's at the bottom of the screen, with antigen and MHC expressed on the antigen presenting cell, shown in yellow at the top of the screen. There are also additional co-receptor molecules such as CD4 or CD8 that are involved in this interaction between the T cell antigen receptor and the MHC. The second signal involves CD28 that's expressed on resting T cells and B7-1 and B7-2 that are on the antigen presenting cells. B7-1 and B7-2 aren't there in resting antigen presenting cell, but as the antigen presenting cells become activated, they express these ligands for CD28, and then you can initiate the second signal or co-stimulatory signal. So in the periphery, you have dendritic cells with a lot of receptors that are able to recognize microbes. The microbes have molecules on them called pathogen-associated molecular patterns, that PAMPs there on the right, and they can interact with a number of different receptors on antigen-presenting cells, including receptors that recognize things like bacterial peptides or endotoxin or DNA or RNA. So in the periphery, the dendritic cells interacts with these PAMPs on the microbes, takes up the microbes, gets stimulated by the PAMPs, migrates through the lymphatics to the lymph nodes. And in the lymph nodes, now they're quite a different cell. They are expressing a number of adhesion molecules such as ICAM1 and ICAM2. They have B7, the ligand for CD28 on their surface, and they're also secreting cytokines that attract T cells. And then the T cells interact with the dendritic cells and become activated. This is just one of the first papers that looked at the power of this dual signaling, where these are lymphocytes, T cells that were stimulated either by just anti-CD3 alone, which binds to and activates the T cell antigen receptor, or they are stimulated by anti-CD28 that activates the co-stimulatory molecule signal 2. So you can see with signal 1 alone or signal 2 alone, you don't get very much, but when you have both of those signals, you get a tremendous activation of T cells. In this case, they're measuring proliferation, but you could also look at IL-2 production or expression of the IL-2 receptor on the T cells. So what happens if you only get one of these signals? If you only get signal 2, which is a co-stimulatory signal, in other words, you're a T cell that is come into the lymph node and it's interacting with some activated dendritic cells, but they don't have the antigen to which you're specific, nothing much happens. You have interaction with the co-stimulatory molecules, but since you're not having signal 1, this isn't really sensed as a positive signal. Conversely, you can also just get signal 1. You can have a non-activated dendritic cell carrying antigen, particularly self-antigen, come into the lymph node where it is not expressing B7-1 or B7-2 on its surface. So you don't get signal 2, but there is antigen in the MHC, and so you do have the ability of T cells to recognize and get that signal. Those T cells that only get signal 1 without signal 2 may become deleted or anergic. They certainly don't become activated. And so in this way, the dendritic cell is really able to control what's going on with the T cell, inducing both activation and promoting peripheral tolerance. So this co-stimulation is really critical in a number of different clinical situations. Shown in white are the things that we want to happen. In other words, if you have antigen and co-stimulation, you get protective immunity and you get tumor immunity. If you have no co-stimulation, then you don't get the induction of allergy to harmless environmental antigens. You get acceptance of a grafted organ, and you don't get autoimmunity. So obviously, you can control a lot of different immunological processes by controlling the co-stimulation. If you promote co-stimulation, then you can enhance protective immunity or tumor immunity. If you block co-stimulation, you may be able to treat allergic disease, graft rejection, and autoimmunity. This is a little bit of a segue. I was going to explain why we have CD4 and CD8 cells. And the reason we have CD4 and CD8 cells is because antigen can be presented either by MHC class II, which would be things like HLA-DR, or it can be presented by MHC class I, which would be things like HLA-A or B. What you have with CD4 is that it forms a co-receptor with the T-cell antigen receptor. In other words, both the T-cell antigen receptor and the CD4 bind to the same MHC class II molecule. The T-cell antigen receptor binds to the specific peptide in the groove of the MHC, and the CD4 binds to a invariant portion of the MHC class II. So you get both the binding affinity of the T-cell antigen receptor and the binding affinity of CD4, and also both of those molecules can give a positive signals to the CD4 T cell. In contrast, CD8 is the molecule that binds to MHC class I. And so when you have a T cell that is CD8 positive, it has a much easier time interacting with antigen in the context of MHC class I. There are big differences in how MHC class I and MHC class II get loaded with antigen. MHC class II is loaded with antigen from the outside of the cell. If you have an external particle that is phagocytosed by a macrophage, it will enter into a phagocytic vacuole with MHC class II and be digested. And then the peptides will actually be loaded onto MHC class II, and that will be routed back to the surface of the cell, where then CD4 T cells can recognize that antigen in the context of MHC class II. Similarly, on B cells, they can take up antigen via the specific antigen on their surface. So anti-tetanus B cells can take up tetanus antigen by their surface immunoglobulin, and then that gets endocytosed and presented again with MHC class II. That gets routed to the surface of the cell, and then the CD4 cells can recognize the antigen in the context of MHC class II. With things like viruses, which are in the cytosol of the cells. MHC class 1 is the critical molecule. You have a whole sophisticated machinery in the cytosol of cells that's able to degrade peptides. This is the proteasome. It degrades peptides into degrades proteins into small peptides, which are then transported into the endoplasmic reticulum, and there they can be associated with MHC class 1. Almost all cells express MHC class 1 except for neurons and erythrocytes. So MHC class 1 presentation is something that all epithelial cells, for example, are able to do. So when you get infected with influenza virus, the epithelial cells can process antigen from their cytosol and express it on their surface and then be recognized by CD8 cytotoxic T cells where the infected cells can then be cleared. So an immune response is very much like a tide where you, and these are the Hopewell rocks up in Nova Scotia that have one of the largest tides in the world. On the left, you can see low tide, where there's a lot of people standing around the bottom of the rocks. At high tide, the water goes up about 25 feet and would have completely inundated those people had they remained there. And then again, the tide goes out, and again, the people come out, and you can walk around the rocks. So with an immune response, you have something very analogous to this. You have to build up your immune response to a crescendo. You also have to make sure it doesn't continue to go up, that it in fact plateaus. And then you have to get it to recede so that you don't continue to have chronic inflammation and have damage to tissue as a result of the adaptive immune response. Some of the adverse effects of overstimulation are shown here, things like cerebral malaria or fulminant hepatitis B, or macrophage activation syndrome, or the reversal reaction you see with lepromatous leprosy when it gets treated with drugs. All of these are examples of overstimulation of the immune system, and this really needs to be controlled. So one mechanism the immune system is controlled is by having a co-inhibitory receptor, and in fact we have many of them, that downregulates the immune response of T cells. So here's, this diagram is showing CTLA-4, which is a molecule on activated T cells that can bind to B7-1 and B7-2 and then prevent the interaction of those ligands with CD28, which ordinarily gives you the second signal for co-stimulation. Here's CD28 and B7-1 shown on the left, and the CTLA-4 and B7-1 shown on the right. And what you can see is that the CTLA-4 has a distinct advantage. It has the capacity to bind multiple B7-1s and form these clusters with a high avidity. And so it will outcompete CD28 and shut off the immune response. If you have a resting T cell shown here in the top, it already expresses CD28, and it also expresses the T cell antigen receptor. If you have a resting antigen-presenting cell, you only get signal 1 through the T cell antigen receptor. But if you activate the antigen-presenting cell, then B7-1 is expressed on the surface, and so you can get not only signal 1, but signal 2. The T cells become activated, they proliferate, they have their effector functions, but then you need to, one of the other things that happens when the T cells become activated is they begin to express CTLA-4, and this now can lead to the inhibition of T cell activation so that the response plateaus and then disappears. This figure shows another co-inhibitory molecule called PD-1, which binds to PD-1 ligand. Again, the PD-1 is on the T cells at the bottom, and the PD-1 ligand is at the top, and this gives an inhibitory signal that doesn't compete for ligand with the CD28, but in fact interrupts the signaling of CD28 in the cytoplasm of the T cells. One of the ways people have figured out how important some of these inhibitory receptors are is by knocking them out in mice. When you knock out CTLA-4 in mice, you get a mouse that only lives a few weeks, and then it dies because of massive proliferation of lymphoid cells and invasion of invasion of organs. Shown in these photomicrographs are the pancreas. On the left side is a wild-type mouse, and you can see a normal pancreas with normal islets. On the right-hand side is the CTLA-4 knockout mice, and the pancreas has been completely destroyed by this infiltration of activated T cells. Another situation where these inhibitory receptors are important is in malaria. In a mouse model of malaria, it's been shown that if you give antibodies against CTLA-4 and PD-1, in other words, if you block the co-inhibitory receptors, you can induce cerebral malaria. So shown on the left is the infiltration of CD8 T cells, which occurs when you block the co-inhibitory molecule CTLA-4 or PD-1. This is what causes the damage. It's not actually the infection, it's the immune response. Another aspect of co-inhibition is that you can develop immunological exhaustion. This occurs when you lose CD8 T cell function. This is seen in a number of different chronic infections and also cancer. Some of these are shown at the bottom here. HIV, hepatitis C virus, or melanoma are all situations where you can have exhaustion of CD8 T cells, so they no longer clear virus or kill the cancerous cells. CD8 T cell exhaustion correlates with having high levels of antigen, with having a chronic, long-duration infection, and also it correlates with the loss of CD4 help. By blocking inhibitory receptors, you can rescue CD8 T cells from this exhaustion. Shown here in this diagram are sort of what happens over time with a chronic infection. That's shown in the diagonal set of cells where labeled inflammation. At the top is what happens when you have a short-term infection, an acute infection where you have the effectors generated. They produce lots of interferon and they're cytotoxic and they can proliferate and they don't apoptose. And then of course they rapidly disappear once the infection is cleared. If the infection continues, then you have T cells that progressively lose effector function. They become unable to lyse other cells. They no longer produce IL-2 or interferon. And they develop, they express on their surface a number of inhibitory receptors such as PD-1 and become much more susceptible to apoptosis. So what people are now beginning to look at is whether or not you can give antibodies against PD-1 and other co-inhibitory receptors and reestablish function. So shown in the middle is sort of the spectrum of CD8 T cells in a chronic infection, where you have some that are functioning fairly well and others that are really not functioning at all and express very high levels of PD-1. If you give an antibody against PD-1, what you find is you rescue the cells with an intermediate level of expression of the PD-1 on their surface. So in other words, cells that are marching down this pathway of exhaustion but haven't really hit the bottom can be rescued with just the anti-PD-1 antibody. And in animal models, they're now showing that this can help with chronic infections, such as immunodeficiency virus. What is hoped in the future is that you can combine anti-inhibitory molecules against PD-1 or against CTLA-4 with additional stimulators to rescue even a higher number of these cells and treat not only chronic infections such as hepatitis C and HIV, but also begin to treat some of the tumors that might be susceptible to immune stimulation. So in conclusion, co-stimulation is critical for determining whether or not an immune response is induced, and the key molecule for co-stimulation is CD28, interacting with B7-1 and B7-2 on the surface of cells. If you have inadequate co-stimulation, you can have weakened host defenses leading to infection or cancer. If you have inappropriate co-stimulation, you can have allergies, autoimmunity, and graft rejection. Co-inhibition is crucial for putting the brakes on immune responses. CTLA-4 and PD-1 are some of the critical molecules involved in co-inhibition. If you have inadequate co-inhibition, you can develop autoimmunity or autoinflammatory diseases. If you have inappropriate co-inhibition, you can also develop immunological exhaustion in the setting of chronic infection and cancers. There are now approved therapies that inhibit co-stimulation. One of these is CTLA-4 Ig, and this has been approved for both autoimmune diseases and transplantation. There are also therapies that inhibit co-inhibition, and these have been approved for malignancies, namely anti-CTLA-4. And they're now being tested in some chronic viral infections. Given the large number of molecules involved in co-stimulation and co-inhibition, it's really very likely that we're going to be seeing a lot of therapies that are targeting these pathways, and these will be used not only in autoimmune diseases but also in infection and oncology. So this concludes our talk. It's really been a delight to be here at the Cleveland Clinic today. Thank you very much for your attention.